Thanks for the introduction. And as you probably gathered, we had the hard word from Russell about keeping to time, so I'm going to have to go through this pretty quickly. So if there's something that's not quite clear, I'm perfectly happy to speak about it a bit later. And what I want to do today in this presentation is really give you some summaries about the work we've been doing with gene editing at Scion, how we're going to use this in the Winning Against Wilding project, and then, since it's a challenge session, talk a bit about some of the challenges we're facing and a public survey we've done to look at some of the challenges around using biotechnology. And just firstly, I wanted to reiterate to you, you've probably heard this before, I think it was spoken about last year, what the differences are between transgenic modification, which you probably know as GE or GM, and gene editing. And you've probably heard of CRISPR because that's the most common form of gene editing, but it's certainly not the only one. And as you can see on the slide, the main difference is with transgenic modification, the key factor is that you add new genetic material. So you take genes or DNA from one individual and you put them into another. And this is a very powerful technology because it allows you to make transformational changes. So you can add a trait that doesn't exist in the host. And it's widely used in agriculture, medicine, and increasingly in sort of industrial biotechnology and the green economy. However, certain versions of gene editing you can do without adding this new DNA. You can basically persuade the host organism to make a very precise change to a gene without you adding any DNA. So it's rather like a directed mutagenesis. You can select a gene you want to change, and you can get the host organism to change it. And really the main advantage of this is that these changes can stop genes working. And so if you can get an interesting phenotype or a useful phenotype by turning off a gene, you can use gene editing to do this. This just goes through the stuff I've just said, so I won't repeat the first bit. I'll reiterate that it's very precise. You can actually select a particular place in the genome. You can go to this place, make a change, and have no other effect on the host. And importantly, the sort of changes you make can't be distinguished from the changes you get either in evolution through spontaneous mutation or if you use mutagenesis, a technique which has been widely used in plant breeding and isn't regulated. And for these reasons and others, as I'm sure you've heard before, many places decided how they're going to regulate gene editing where you don't add new DNA, and they've decided they're not going to regulate it as a GMO. They're going to regulate it in the same way as any other breeding technology. You can see the list of countries which have sort of come to this decision, and Australia recently decided that this was how they were going to do regulation. And China isn't actually on that list, but it looks like they're going to make the same decision. As I'm sure many of you know, New Zealand has decided, alongside Europe, that it's going to regulate this technology as a GMO. So that's currently a bit of a barrier I'll say a little bit more about later. And I guess principally because this technology isn't being regulated as a GMO, it's meant that products are coming to market very quickly. Just 7 years after the technology was developed, the first products went on sale earlier this year. And they're not burdened by the huge regulatory costs of GMOs. It's usually assumed that bringing a GMO to market costs $100 to $150 million or US dollars in regulatory fees. And so it's sort of driven these products into these industrial Monsanto-type organizations. But this new lower cost thing is allowing niche products and more targeted things to come to market. And I just wanted to give you a little update. Due to time, I won't go through this very much, about where we've got to with gene editing over at Scion. And obviously we see tremendous potential in gene editing, so we've been working on it for a couple of years, trying to get it to work in conifers. And over the last few months, we've actually managed to carry out our first few gene editings. So we've edited 3 or 4 genes, so we're pretty confident we can actually do this with any gene we select, given that we can design the proper construct to do it. And I won't go through the details of this slide, other than I wanted to use it to show you what I meant by a very precise and targeted approach. And this little bit of sequence you can see in the middle, these AGTCs, are just a little bit of DNA sequence of a gene. And we wanted to stop that gene working. So we designed one of these CRISPR elements to target the place where the arrow's pointing. And then we did some gene editing and got some individuals, and we got the sequence from one of them, which I've shown below. And you can't actually probably see the difference unless I circle it. But in the wild type or unedited plant, we actually have a sequence of ACA. In one of our edited plants, there's a very small change. The plant has actually inserted an extra C, so a very minor change. So it's now ACCA, and it's precisely at the place we directed it to actually happen. And I should have said that the gene above produces a protein, which is the thing that actually does the work, that looks like this. And the prediction with our edited protein is it'll look like this, which means it'll be non-functional, so we should be able to investigate what that gene does. And we're actually now growing up some plants, because we do this in tissue culture, to actually see if this actually is having a phenotype. And I forgot to say this was in Pinus radiata, but we've recently also done it in Douglas fir. And if you look at the literature, I said a lot of things coming to market, and the sort of things which agricultural companies are bringing to market are the sort of traits you can see here. So some of these have obvious parallels to what we could do in forestry. So it is certainly something in the future that could be very valuable. And the bottom thing I have here, sterility, leads me nicely in to the Winning Against Wildings programme. And for those of you who don't know about the Winning Against Wilding programme, it's an MB and Forest Levy funded project looking at all aspects of wildings, from how they generate, control, prevention, so it encompasses the whole area. And part of the work being funded at Scion is to try and use biotechnology to actually make Douglas fir unable to produce wildings. And the reason we want to do this is because Douglas fir is a potentially lucrative crop in the South Island, but because it's very prone to generate wildings, there's a lot of pressure not to plant it. And really what we want to do is pretty simple, or simple in theory at least, is we want to use either transgenic technology or the gene editing I showed you earlier to find some genes which are necessary for reproduction, so most likely cone-related genes, and inactivate these. And through this, we would stop reproduction. And this slide is just to show you that there's nothing new in doing this. Man has been using sterile plants for many, many years, going back to the banana, where a sterile hybrid was domesticated 7,000 years ago, right through to lots of citrus fruits, which have been bred to be seedless or sterile using mutagenesis. So it's pretty much along the same trail as what's gone before. And where have we got with this? We started this project about 3 years ago, before we had actually got gene editing working. So our first efforts were in making transgenic trees, and we've now got several different, potentially sterile transgenic trees growing on our GMO glasshouse, or contained GMO glasshouse. This is regulated by the EPI and MPI, so we can only grow these in containment indoors. And we've also got additional gene-edited trees in the pipeline on the way. So this session was about challenges, so I thought I'd throw in what our biggest challenge is, because we've suggested that we can produce these trees, but we need to test them. And in New Zealand, that's exceptionally difficult because of the Has No Act and the regulations surrounding how you can use GMOs. And what we really need to do to test these trees is grow them outside so we can see that they grow normally with our modifications and check that they are indeed sterile. But under the Has No Act, you can't actually grow trees outside and let them reproduce. You have to kill them before they reproduce. So it's a bit of a challenge to be able to do that. And it's not just a challenge for forestry and Scion, it's a challenge for the whole primary sector. Ag Research have got some ryegrasses which potentially have climate change capabilities, or preventing climate change, mitigating it, but they're not able to grow these in New Zealand because ryegrass reproduces, so they have to doing it overseas. And this is increasingly becoming a problem with gene editing because lots of people want to use this technology. It looks like many overseas markets won't be regulating it, but you can't test products in New Zealand because of this decision that gene editing will be a GMO in New Zealand. And so really a lot of work's going on around how to overcome this challenge. And you've probably seen in the press over the last few months, there's been a lot of activity around, is it time to have a debate about these technologies? I think the Prime Minister's science advisor, who was here yesterday, has actually been vocal about this a few times. The Royal Society are certainly encouraging it. And there's been lots of interaction with government by various agencies to try and do this. And it seems as there is some progress, but where that goes, we don't know yet. Is that the 1 minute or 5? 5, great. And so towards doing this, what we really need to do is find out what the New Zealand public think and engage with the public around how we can actually have this discussion. Do we need to have a debate about whether we use these technologies or whether New Zealand doesn't use them, and potentially risks falling behind our competitors who are using these technologies? And towards this, Scion recently commissioned a survey to look at public perceptions around using these biotechnologies, particularly with regard to forest trees, and also with a focus on using them for things which might have positive environmental impacts. And you can see the survey that Colin Brunton carried out very recently. It finished back in August, so we haven't done too much analysis of this. I'm just going to present you with some of the initial data which Colin Brunton provided to us. But you can see it's a pretty big survey, about 4,000 New Zealanders responded, and we've got pretty good demographics. We've captured the various groups within New Zealand and the different regions. And if we're going to have a debate around whether we actually use these technologies, one of the things we need to do is see how informed or knowledgeable people are about these things, because clearly you can't have a debate if people don't know. And so one of the first questions we asked people was actually how aware they were of some different biotechnologies. And by aware, we meant they'd heard of them. They knew what they were. Not that they knew what they were, sorry. And you can see from looking at the slide that people were pretty aware of GMOs, which is unsurprising. It's in the press a lot. And if you read social media, it's not hard to find some sort of view on it. And gene editing also, quite a lot of people have heard of it. It's also been in the press a lot, and so I guess that probably explains that. Next, we wanted to know not how aware people were, but actually how knowledgeable they were. Did people actually know anything about this technology? Because obviously knowledge is what's needed if people are going to have an informed or be able to make an informed decision. And the answers we got here, you can see. Are that clearly people aren't very knowledgeable about these technologies. And this was people assessing themselves, saying whether they were knowledgeable. And you can see that for both genetic modification and gene editing, there were very low levels of knowledge. And if you look at the bottom, you can actually see the figure for the people who said they were aware. And even amongst the people who are aware, they actually have very low knowledge. This is a really big challenge if there's going to be a debate where people can give a view based on evidence. And it's the same story, I guess, all over the world, that there is this lack of knowledge. And quite how to address that is, I guess, one of the really big challenges. As I said, the Royal Society are doing this. Lots of scientists go out and do this, but I guess it's up to everybody who has an interest in seeing these technologies used to actually go out and try and inform the public about this. But quite how this can happen is extremely difficult. And finally, at the end of the survey, we actually presented some different scenarios around using gene technologies. We gave a little description of what the problem was and how we might use technology to provide a solution. And then we asked people whether they found this acceptable or not acceptable. And the three scenarios we presented were a theoretical one around using gene editing as a possible solution for Kauri dieback. We then presented a transgenic technology option, which was something that's actually happening in the USA, where they're using transgenic techniques to try and save an iconic species called the American chestnut, which has been pushed to the edge of extinction by a blight disease, and they're actually currently doing some field trials with transgenic trees to solve this. And we also asked them about wildings and whether if we could gene edit Douglas fir so that it didn't add to the wilding problem, they would find this acceptable. And what you can see if you look at the results we got from all of these, that in all cases people were very accepting of these technologies, probably much higher than we thought based on the sort of social media news you hear. And the people who were totally opposed was actually very, very small. Those who found it not at all acceptable was really a tiny minority. So this really does suggest that the New Zealand population is at least open to having a debate on this, and we need to find a way to communicate them so that they can have an informed debate. And just to finish, just to summarise what I've said, we've developed gene editing at Scion in conifers in both radiata and Douglas fir. This is a lot of work to get it working much better than it currently does, and we've started to use this in the Winning Against Wildings project. And we're also working towards how we might be able to test these trees in New Zealand, really through working towards having a debate where the government might decide that the 20-year-old Hasno Act is no longer fit for purpose, which is, in many people's opinions, the sort of the status of it. That's me. Thank you very much.